A brief correction before this begins, since it matters for accuracy the same way it has mattered in related reconstructions covering this same distant, cold, and frequently misunderstood chapter of human evolutionary history. The best supported dating evidence places the disappearance of the last Neanderthal populations at approximately 40,000 years ago rather than 30,000, with a handful of southern Iberian sites possibly extending a few thousand years beyond that at the outside, and several once famous younger dates from Gibraltar and southern Spain now considered unreliable following more rigorous redating published in Nature in 2014. What follows describes the diet of Neanderthal populations across the final tens of thousands of years of their existence, built from isotope chemistry, dental calculus, coprolites, and butchered bone recovered from sites spanning from Gibraltar to Siberia. And the picture that emerges is considerably stranger, more regionally varied, and more scientifically contested than the simple label of Ice Age meat eater has traditionally suggested. A picture this reconstruction will trace site by site, region by region, and food source by food source. It is also, at its core, a story about survival math, about exactly what kind of food a body built. The way a Neanderthal body was built actually needed to get through a glacial winter alive, and about how much ingenuity, regional adaptation, and outright experimentation that survival math ended up demanding from populations spread across an enormous and wildly varied stretch of Ice Age Eurasia, from the mild Mediterranean coastline of Gibraltar to the harsh continental interior of southern Siberia. For decades, the dominant scientific picture of Neanderthal diet rested on a single, powerful, and genuinely startling type of evidence: stable isotope analysis of nitrogen preserved in fossilized bone collagen. When an animal eats, the ratio of heavy to light nitrogen isotopes in its food gets passed into its own tissue with a small, predictable upward shift at each step of the food chain, meaning herbivores carry a certain nitrogen signature. The carnivores that eat those herbivores carry a slightly higher one, and so on up the chain. When researchers began measuring this signature in Neanderthal bone collagen recovered from sites across Europe, the results were startling. Neanderthal nitrogen values consistently matched, and in some cases exceeded, those of the most committed carnivores sharing their ecosystem, animals like wolves and hyenas that derive the overwhelming majority of their diet from meat and almost nothing else. Detailed isotopic work led by researchers including Michael Richards and Eric Trinkhaus concluded that Neanderthals across their European range were obtaining nearly all of their dietary protein from animal sources, occupying the very top of the regional food web, a position described in the scientific literature as functionally equivalent to a hypercarnivore, a term normally reserved for predators whose diet is at least 70% meat, and one that placed Neanderthals on paper in roughly the same dietary category as lions, rather than anywhere close to the broad, flexible omnivory typical of most primates, including modern humans. The animals actually being hunted to produce this signature were, at most well-studied European sites, the large herbivorous mammals that dominated Ice Age landscapes. Analysis of butchered bone assemblages recovered from Neanderthal occupation layers across the continent shows a consistent focus on medium to large ungulates, reindeer, red deer, horse, bison, and aurochs, supplemented in certain regions and periods by genuinely massive prey. At the site of Neumarkt Nord in Germany, researchers have documented evidence that a local Neanderthal population hunted the straight-tusked elephant, an animal considerably larger than any modern elephant species, standing well over 4 meters at the shoulder, in the largest individuals, on a surprisingly regular basis across the roughly 125,000-year-old occupation layers there, perhaps once every 5 to 6 years, a hunting achievement that would have required substantial coordination among the members of a family or community group, and that would have provided in a single successful kill. an amount of meat, fat, and marrow capable of sustaining many individuals for a considerable stretch of time, quite possibly weeks, depending on how effectively the group was able to preserve or otherwise make use of a carcass that size before it spoiled. Isotopic values from the Chagarskaya cave, Neanderthals of southern Siberia, dated to roughly 54,000 years ago, tell a broadly consistent story from an entirely different corner of the Neanderthal range, with researchers identifying horse and bison as the most likely primary prey species based on comparing the Neanderthal isotopic signature directly against the isotopic values of the surrounding fauna, and finding nitrogen values roughly 5 per mil higher than the local carnivores, and 7 per mil higher than the local herbivores, a genuinely extreme trophic position by any measure, confirming that this specific dietary pattern, heavy reliance on the largest available terrestrial herbivores, held true even at the far eastern edge of the species' known geographic range, thousands of kilometers from the European sites where the pattern was first documented. For a long time, this consistent hypercarnivore level nitrogen signature was read essentially at face value, as direct proof that Neanderthals were obligate, near-exclusive meat eaters, an interpretation that fed directly into broader theories about why the species eventually went extinct, since a population locked into such a narrow dietary strategy would have been considerably more vulnerable to the collapse of large game populations during periods of rapid climate change than a more flexible, omnivorous competitor. But more recent research has begun to complicate this picture in a genuinely significant way, not by disputing the underlying isotope measurements themselves, but by questioning whether meat consumption alone is actually sufficient to explain numbers this extreme. A study published in the journal Science Advances proposed an alternative, or at least a supplementary explanation, for the unusually elevated nitrogen values recorded across the Neanderthal fossil record, pointing to a detail of hunter-gatherer food storage that had previously been largely overlooked in the isotope literature: the consumption of maggots. Researchers studying ethnographic records of recent hunter-gatherer populations who store meat and fish for extended periods found that these stored foods routinely become infested with fly larvae, and that these maggots themselves are frequently and deliberately eaten, sometimes in substantial quantities, as a normal, unremarkable part of the diet, rather than an unfortunate contaminant to be avoided, a detail that modern readers accustomed to refrigeration and food safety regulation tend to find viscerally surprising, but that shows up repeatedly and matter-of-factly across ethnographic accounts of hunter-gatherer groups who regularly cache meat for later use. Critically, maggots feeding on decomposing meat carry their own further elevated nitrogen signature, layered on top of whatever signature the meat itself already carried, meaning a diet that included regularly eating maggot-infested stored meat could plausibly push an already meat-heavy nitrogen signature up into the same range researchers have been interpreting as evidence of essentially pure, unmixed carnivory. This does not mean Neanderthals were not eating substantial quantities of meat, since every other line of archaeological evidence, from butchered bone to hunting technology, confirms they clearly were. It means the specific claim that their diet was as exclusively extremely carnivorous as the raw isotope numbers alone suggest may be, at least in part, an artifact of how that meat was being stored and consumed rather than a complete and literal description of what made up their plates. A reminder that even a measurement as seemingly objective and precise as an isotope ratio still requires careful, informed interpretation before it can be translated into an accurate picture of an extinct population's actual lived diet. This growing recognition that the isotope evidence, powerful as it is, tells an incomplete story on its own, has been reinforced by an entirely separate and increasingly rich body of research examining a source of dietary evidence isotopes cannot capture at all: dental calculus, the hardened mineral deposit that builds up on teeth during life, and that, once fossilized, can trap and preserve microscopic plant remains for tens of thousands of years. A landmark 2011 study led by Karen Hardy examined dental calculus recovered from Neanderthal individuals at 2 sites representing dramatically different environments and time periods, Shanidar III in Iraq and Spy-Rione II in Belgium, and found direct physical evidence of plant consumption at both locations in the form of preserved starch grains and phytoliths, the microscopic silica structures that form inside many plant tissues and survive essentially indefinitely once deposited. The specific plants identified differed meaningfully between the 2 sites in ways that closely tracked their very different local environments. At Shanidar, the recovered plant remains matched species already known from soil samples and other botanical evidence at nearby Near Eastern Neanderthal sites, including date palm and various legumes well suited to the warmer, more Mediterranean-influenced climate of northern Iraq, while the material recovered from Spy showed clear evidence of underground storage organs, roots, tubers, and similar starchy plant parts of exactly the type researchers had already predicted European Neanderthals. Occupying colder, more northern environments would likely have relied on as a stored, reliable carbohydrate source through the leaner months of the year, when fresh above-ground vegetation would have been buried under snow or simply dead for the season. Crucially, several of the starch granules recovered from both sites showed damage patterns consistent with having been cooked, direct physical evidence that these plant foods were not simply gathered and eaten raw, but were being deliberately processed with heat, a level of food preparation sophistication that pushes back considerably further in time than many researchers had previously assumed for this specific behavior, and that carries real nutritional significance since cooking starchy tubers substantially increases the proportion of their carbohydrate content that a human or Neanderthal digestive system can actually break down and absorb compared to eating the same tuber raw. The same broad research team returned to the Spy material with a different analytical approach in a follow-up study, examining the dental calculus not just for plant microfossils, but for preserved biomolecules and microbial DNA. And the results, published under the memorable title Neanderthal Medics, revealed something considerably more specific than simple plant consumption. The Spy Neanderthals appeared to have consumed woolly rhinoceros and wild sheep, a genuinely meat-heavy profile consistent with the broader isotopic picture for northern European populations, alongside evidence of consuming mushrooms, which themselves would have offered a further, largely overlooked source of dietary protein and micronutrients within an otherwise meat-dominated regional diet. At El Cedron in Spain, by sharp contrast, the same kind of analysis found essentially no meat signature at all in the dental calculus, but did find evidence of pine nuts, moss, mushrooms, and other plant material, along with, more strikingly, chemical signatures the research team interpreted as consistent with self-medication, compounds matching those found in the bitter, medicinally active plants, yarrow and chamomile, identified in the calculus of an individual whose skeleton showed evidence of a painful dental abscess, along with signs of an intestinal parasite infection. A genuinely striking possibility that this specific Neanderthal may have been deliberately consuming bitter, pharmacologically active plants to manage the symptoms of illness rather than for any nutritional value alone, since neither yarrow nor chamomile offers meaningful caloric content and both taste, by any reasonable human standard, unpleasant enough that their consumption is difficult to explain as ordinary. Foraging, an interpretation that, if correct, would place this specific El Cedron individual among the very earliest documented examples anywhere in the hominin fossil record of deliberate, targeted self-medication using plant compounds for their pharmacological rather than nutritional properties. This stark contrast between the heavily meat-focused Spy population and the far more plant and fungus-oriented El Cedron population recovered using the same analytical methods applied to the same type of preserved material, stands as some of the clearest available evidence that Neanderthal diet was not a single, uniform strategy applied identically across their entire geographic and temporal range, but instead varied substantially based on local environment, available resources, and quite possibly distinct cultural or group-specific food traditions passed down within particular communities, echoing the same pattern of regionally specific, family-transmitted practical knowledge already well documented in Neanderthal stone tool traditions. A broader review of dental calculus evidence, examining samples from 5 separate archaeological sites spanning the northern Balkans and the western, central, and eastern Mediterranean, confirmed this same underlying pattern, finding that despite a persistent scientific narrative describing Neanderthal diet as heavily meat-based and broadly similar across environments, the plant micro-remains preserved in dental calculus revealed a genuinely varied and, in the researcher's own description, widespread use of non-animal foods, including a range of starchy plants, indicating that plant consumption was not a rare marginal supplement to an otherwise carnivorous diet, but a stable, consistent, and geographically widespread component of the broader Neanderthal dietary niche, present across environments ranging from the cold, continental interior of the Balkans to the warm, dry coastlines of the eastern Mediterranean. Coastal and near-coastal Neanderthal populations add yet another distinct dimension to this increasingly varied picture, one documented particularly well at the cluster of cave sites in and around Gibraltar. Excavations there have recovered butchered remains from 143 different bird species, a genuinely remarkable degree of avian dietary diversity, including numerous ground-dwelling species such as the common quail, corncrake, woodlark, and crested lark, animals that would have required a very different hunting approach, patient low-risk trapping, rather than the coordinated pursuit needed for large game, from the rest of the Neanderthal diet documented at inland sites. At Vanguard Cave specifically, researchers have recovered direct evidence that Neanderthals were consuming dolphin and Mediterranean monk seal, genuine marine mammal prey exploited by a species traditionally associated almost entirely with terrestrial hunting, evidence that at least some coastal Neanderthal populations were willing and able to incorporate marine resources into their diet when the local geography made that resource available, whether through active hunting of stranded or vulnerable animals or through opportunistic scavenging of carcasses washed ashore, a question the bone evidence alone cannot fully resolve. This coastal dietary flexibility fits into a broader pattern. Researchers studying Iberian Neanderthal sites have increasingly documented evidence of shellfish consumption and other marine resource use at multiple locations along the Mediterranean and Atlantic coastlines of the Iberian Peninsula, a set of behaviors that had, for a considerable stretch of the 20th century, been assumed to represent a distinctly modern human innovation rather than something Neanderthal populations were independently practicing tens of thousands of years earlier, a finding that has contributed directly to the broader scientific reassessment of Neanderthal cognitive and behavioral sophistication that has unfolded steadily across the past 2 decades of research. Direct physical evidence of what Neanderthals actually swallowed and digested, as opposed to indirect inference from isotopes, butchered bone, or trapped dental residue, comes from an even rarer and more specific category of archaeological material, fossilized feces. A study examining coprolites, preserved fecal material, recovered from the Middle Paleolithic site of El Salt in Spain, found direct chemical evidence in the form of specific biomarker compounds known to derive from digested plant sterols alongside cholesterol from digested animal fat and protein, confirming that the individual or individuals who produced this particular sample had consumed a genuinely mixed diet combining both animal and plant material. Physical proof recovered from the actual digestive waste of a Neanderthal That omnivory rather than strict carnivory characterized at least this specific population's actual realized diet, though the same analysis noted a lack of clear indicators for the consumption of starchy tubers specifically at this particular site. A reminder that even within the broader category of documented plant consumption, the specific plants being eaten continued to vary considerably from one Neanderthal population and location to the next. Precisely the kind of fine-grained regional variation the isotope data alone, working only at the level of broad meat versus plant protein ratios, could never have revealed on its own. The Chagirskaya cave material from southern Siberia offers one further, particularly well-documented case study in how researchers now combine multiple independent lines of evidence to reconstruct diet at a single site, since the same population whose isotope values indicated a heavily meat-based, high trophic level diet built primarily around hunting horse and bison also showed direct evidence of plant consumption in their dental calculus, with researchers recovering 32 separate phytolith specimens across the sampled individuals, alongside a single preserved starch grain, plant material present in genuinely small quantities compared to the dominant meat signal, but present nonetheless, confirming that even this far eastern, isotopically extreme Neanderthal population was not living on a diet of meat alone, even if plants clearly represented a minor, rather than a central component of what they were actually eating on a typical day. Researchers studying this specific site have noted that the comparative rarity of plant microfossils at Chagirskaya, especially set against the far richer plant evidence recovered from Western European and Near Eastern sites like Spy and Shanidar, may reflect the genuinely more limited range of edible plant resources available in the colder, more continental climate of the Altai Mountains during this period, rather than any fundamental difference in Neanderthal dietary behavior or willingness to eat plants when they were actually available, a distinction that matters considerably for how this evidence should be interpreted, since it separates a population's underlying dietary flexibility from the simple external constraint of what their specific local environment actually had to offer them in any given season. Underlying all of this dietary evidence, and helping to explain why meat consumption, whatever its precise proportion relative to plants at any given site, remained such a consistently dominant feature of the Neanderthal diet virtually everywhere researchers have looked, is the sheer scale of the caloric and nutritional demand these bodies would have placed on whatever food supply was available to them. Researchers modeling Neanderthal energetics, drawing on skeletal evidence of unusually high muscle mass, robust bone structure, and a body plan broadly adapted for conserving heat in cold environments, have estimated that an active adult Neanderthal living through a genuinely cold glacial season would have required a daily caloric intake substantially higher than that needed by a modern human of comparable size, with some estimates placing total daily energy expenditure for an active individual in cold conditions well above 4,000 kilocalories, and considerably higher still for pregnant or lactating women, and for individuals engaged in the kind of sustained, physically demanding activity that hunting large, dangerous prey like horse, bison, or an occasional straight-tusked elephant would have required. Meeting this kind of elevated, sustained energy requirement reliably, across every season including the depths of an ice age winter when plant foods would have been scarcest and least accessible, made calorie-dense animal fat and protein an almost unavoidable dietary priority for any population living in these environments, regardless of how much additional dietary variety plants, fungi, marine resources, or even the deliberately consumed maggots described earlier in this reconstruction might have contributed on top of that essential meat-based caloric foundation. This same energetic pressure likely explains why marrow extraction appears so consistently across Neanderthal butchered bone assemblages, with long bones frequently found deliberately broken open in a manner consistent with extracting the fat-rich marrow within, a genuinely calorie-dense resource that would have offered considerably more usable energy per unit of effort than lean muscle meat alone, particularly valuable during leaner seasons when overall prey availability and body condition, and therefore the fat content of any given kill would have been at its lowest. A period recent hunter-gatherer research has termed the risk of protein poisoning or rabbit starvation a genuine physiological danger that arises when a population relies too heavily on lean, fat-depleted meat without a sufficient parallel source of dietary fat, since the human and Neanderthal liver can only safely process a limited proportion of daily calories as protein alone, meaning marrow, and quite possibly some of the plant and fungal foods documented across this reconstruction would have served a genuinely essential nutritional function beyond simple caloric supplementation. Cooking itself, a behavior with direct implications for how efficiently any of this food, plant or animal, could actually be digested and converted into usable energy, is now documented archaeologically at multiple Neanderthal sites through a combination of hearth evidence and, as already described, the charred, heat-damaged starch granules recovered directly from dental calculus. Cooking meat and plant foods alike measurably increases the caloric energy an animal digestive system can actually extract from them, breaking down tough plant cell walls and denaturing proteins in ways that reduce the metabolic cost of digestion itself, meaning the cooking behavior documented at sites from Shanidar to Spy was not simply a matter of taste or food safety, but represented a genuine, measurable energetic advantage for a population already operating under the kind of elevated caloric demand described above, extracting more usable energy from the same raw quantity of food than an equivalent amount of raw, unprocessed material could have provided. An advantage researchers studying the broader evolution of human and hominin diet regard as significant enough that some have proposed cooking itself as one of the specific behavioral innovations that allowed increasingly large, energy-hungry hominin brains to be sustained on a realistic, achievable daily food supply in the first place. The hunting technology and tactics that made this consistent, large game-centered diet possible deserve attention in their own right, since acquiring calorie-dense meat from animals as large and dangerous as bison, horse, or an occasional straight-tusked elephant was never simply a matter of encountering prey and killing it. Neanderthal hunting weapons, primarily heavy, thrusting spears tipped with carefully shaped stone points, required hunters to approach prey at close range, a strategy confirmed by detailed analysis of impact damage on recovered spear points, along with skeletal trauma patterns documented across numerous Neanderthal individuals. Healed fractures concentrated in the head, neck, and upper body, in a pattern strikingly similar to the injury profile documented among modern rodeo performers who work in close, repeated physical contact with large animals. This close-range hunting strategy carried genuine physical risk, and researchers studying Neanderthal skeletal trauma have proposed that the sheer frequency of these healed injuries reflects a hunting method fundamentally different from and considerably more dangerous than the longer-range projectile hunting that would become more common among later Upper Paleolithic modern human populations equipped with spear throwers and eventually bow technology. Successfully bringing down large, dangerous prey at close range using this kind of weaponry would have depended heavily on group coordination, multiple hunters working together to corner, tire, or otherwise disadvantage an animal before delivering a killing blow. Cooperative behavior that connects directly back to the small, kin-based family and community structures already documented through the genetic evidence recovered from sites like Chagirskaya. Since a hunting strategy this dangerous and this dependent on coordinated teamwork would have functioned best, and perhaps only been survivable at all, within a group whose members knew and trusted one another intimately. Exactly the kind of stable, multi-generational family band the genetic record has independently confirmed these populations actually lived in. Seasonal variation in diet, though harder to document directly than the broad regional patterns already described throughout this reconstruction, can be partially reconstructed by combining what is known about the breeding and migration patterns of key prey species with the specific archaeological evidence recovered at individual sites. A reconstruction that, while necessarily more speculative than the direct physical evidence discussed elsewhere in this piece, still rests on a reasonably solid foundation of comparative ecological data. Reindeer, one of the most consistently hunted species across the northern portion of the Neanderthal range, undertake substantial seasonal migrations even today. And researchers studying Neanderthal occupation patterns at sites positioned along known migration corridors have proposed that at least some Neanderthal groups timed their own movements and hunting activity to intercept these herds during their seasonal passage. A strategy that would have concentrated the most reliable, highest yield hunting opportunities into specific predictable windows of the year, rather than requiring hunters to search for scattered, unpredictable prey across every season equally. Plant foods would have followed an even more pronounced seasonal rhythm, with fresh, above-ground vegetation, fruits, and greens available primarily during the warmer months, while the starchy underground storage organs documented in the SPY dental calculus, roots and tubers that plants use specifically to store energy through their own dormant winter period, would have remained accessible, at least in principle, even after the harshest cold arrived, provided Neanderthal foragers retained the knowledge of exactly where to dig for them beneath frozen or snow-covered ground. A piece of specific, place-based ecological knowledge that, much like stone tool manufacture, would most plausibly have been transmitted within families across many successive generations occupying the same general territory. Comparing this reconstructed Neanderthal dietary strategy against what is independently known about the diet of contemporary anatomically modern human populations sharing parts of the same late Pleistocene landscape offers one further useful angle on this material. Since the two populations' overlapping presence across large parts of Europe and the Near East for many thousands of years means their dietary strategies were, at least potentially, in direct competition with one another for some of the same prey and plant resources. A competitive overlap researchers studying this period regard as an important, if still incompletely understood, factor in the broader story of how these two closely related human populations came to share, and eventually stop sharing, the same landscape. Isotopic studies of Upper Paleolithic modern humans, occupying broadly comparable environments to late Neanderthal populations, have generally found similarly elevated nitrogen values, though researchers studying these values have proposed that modern human populations may have achieved comparable trophic level signatures through a somewhat different combination of strategies, including greater use of freshwater fish and other aquatic resources than most Neanderthal populations are currently documented as exploiting, alongside the broader, more extensive social and trade networks already established through the genetic and archaeological evidence discussed in relation to Neanderthal family structure. Networks that may have allowed modern human groups to buffer local resource shortfalls more effectively by drawing on a wider, more interconnected web of neighboring communities than the smaller, more isolated Neanderthal bands typically maintained. Some researchers studying the eventual replacement of Neanderthal populations by modern humans across Europe have proposed that this difference in the breadth and flexibility of resource networks and exploited food types, rather than any fundamental gap in intelligence, hunting skill, or basic dietary sophistication, both of which the evidence assembled throughout this reconstruction suggests Neanderthals possessed in genuinely impressive measure, may have provided modern human populations with a meaningful, if incremental, survival advantage during the increasingly chaotic, unpredictable climate swings that characterized the final tens of thousands of years before Neanderthal populations disappeared from the fossil and genetic record entirely. A subtle demographic and dietary edge accumulating slowly across many generations, rather than any single decisive competitive advantage visible within any one individual human or Neanderthal lifetime. What all of this evidence, taken together across isotope chemistry, dental calculus, coprolites, hunting trauma patterns, and comparative analysis with contemporary modern human populations, ultimately demonstrates is that the question posed by this reconstruction's title, what Neanderthals actually ate to survive the Ice Age, does not have a single tidy answer, and that this absence of a single simple answer is itself the most important finding the last 2 decades of dietary research on this. Species have produced a Neanderthal in Belgium, gnawing marrow from the shattered long bone of a woolly rhinoceros beside a fire built to cook a cache of wild tubers dug from beneath the frozen ground, was solving the same basic survival problem, obtaining enough reliable, calorie-dense food to sustain a large, active, cold-adapted body through an Ice Age winter, using genuinely different specific tools and resources than a Neanderthal in coastal Gibraltar, trapping ground-dwelling birds and butchering a stranded dolphin on a Mediterranean beach, or a Neanderthal in the Altai mountains of Siberia, tracking horse and bison herds across an open continental steppe, with only the barest supplement of locally available plant material to round out an otherwise near-total reliance on meat. Each of these populations, connected to one another by the same broad Mousterian technological tradition, and as genetic evidence increasingly confirms, by a wider network of kinship and cultural transmission spanning the entire Neanderthal range, nonetheless arrived at genuinely distinct, locally optimized dietary solutions. A pattern of flexible, intelligent adaptation to local circumstance that stands as one of the clearest and most consistently underappreciated pieces of evidence for the sheer behavioral sophistication of a species that, for most of the 20th century, popular imagination was content to picture as a crude, club-wielding brute, concerned with little beyond the next available piece of raw meat, rather than the resourceful, regionally adaptive survivor that decades of patient isotopic, microscopic, and biomolecular research have now conclusively revealed it to have actually been. Neanderthal diet was overwhelmingly, consistently anchored in animal protein and fat, obtained through skilled, cooperative hunting of medium and large game ranging from reindeer and horse to, in certain times and places, genuinely massive prey like the straight-tusked elephant. A dietary foundation confirmed by isotope signatures, matching or exceeding those of dedicated carnivores across virtually every well-studied site on the European and Asian mainland, even if that extreme signature may, in some populations, be partly amplified by the deliberate consumption of maggot-infested stored meat rather than reflecting fresh meat intake alone. Layered on top of that meat-centered foundation, however, was a genuinely wide and, in several documented cases, sophisticated range of supplementary foods and behaviors. Cooked starchy tubers in Belgium, pine nuts and mushrooms and possibly deliberate medicinal plant use in Spain, birds, dolphins, and seals along the coastlines of Gibraltar, phytoliths, and the occasional starch grain even among the isotopically extreme horse and bison hunting population of distant Siberia. All of it processed, in at least some documented instances, through deliberate cooking that would have made every calorie obtained that much easier for the body to actually use. What this evidence ultimately reveals is not a single, rigid Neanderthal diet repeated identically across 50,000 years and thousands of kilometers of profoundly different environments, but a population, or more precisely a network of related genetically connected populations that met the specific nutritional demands of an Ice Age world by combining a stable, calorie-dense core of skilled, cooperative big game hunting with whatever additional regionally specific resources, plant, fungal, marine, or otherwise, their particular corner of that world happened to offer. A flexible, adaptive dietary strategy considerably closer to the sophisticated foraging behavior long associated exclusively with anatomically modern humans than the crude, meat-obsessed caricature that dominated both scientific and popular understanding of this species for most of the 20th century. Fat consumption, specifically, as distinct from lean protein, deserves closer attention in any complete account of Neanderthal survival diet, since the physiological ceiling on how much protein a human or Neanderthal body can safely metabolize without adequate fat intake represents one of the more concrete, quantifiable nutritional constraints researchers can actually model with some confidence. Modern nutritional research has established that the human liver can typically process protein, amounting to no more than roughly 35 to 40% of total daily caloric intake before toxic byproducts of protein metabolism begin to accumulate faster than the body can safely eliminate them, a condition variously described in the ethnographic and medical literature as rabbit starvation or protein poisoning. Named for the historical observation that hunter-gatherers and early explorers surviving on lean rabbit meat alone, without any parallel source of dietary fat, could develop severe illness and even die despite consuming what would otherwise appear to be an adequate caloric intake. For a Neanderthal population whose isotope signature indicates an overwhelming reliance on animal protein, avoiding this specific physiological trap would have depended heavily on prioritizing the fattiest available portions of any successful kill, marrow, brain tissue, and internal organs rich in fat content, over lean muscle meat alone, a prioritization directly reflected in the marrow extraction evidence already discussed throughout this reconstruction, and one that would have made the fat content of a given kill, which varies dramatically with an animal's age, sex, and seasonal body condition, a genuinely critical variable determining whether a successful hunt actually translated into safe, usable nutrition or into a kill that, however impressive in raw meat tonnage, left a hunting party still nutritionally vulnerable. This same fat prioritization logic likely explains a pattern documented at numerous Neanderthal sites, evidence of what researchers term bone grease rendering, the deliberate, labor-intensive practice of crushing already defleshed bone into small fragments and boiling them to extract residual fat trapped within the spongy interior bone tissue, a process requiring the availability of watertight containers, whether animal hide bags, hollowed wood, or some other Ice Age improvisation, capable of holding water over a sustained fire long enough for the extraction to occur. Evidence of this specific practice, identified through the distinctive, highly fragmented pattern it leaves on recovered bone assemblages, appears at multiple sites across the Neanderthal range and indicates that these populations were not simply extracting the easily accessible fat and marrow from freshly killed animals, but were engaged in a considerably more thorough, effort-intensive process of extracting every remaining calorie of fat from a carcass, including fat trapped in bone that a less resourceful or less nutritionally pressured population might reasonably have discarded entirely, a level of dietary thoroughness that speaks directly to just how consistently, seasonally pressing the demand for dietary fat specifically, rather than calories or protein in any general sense, actually was for populations living through the coldest, leanest stretches of the Ice Age calendar. Gut health and digestive adaptation represent one further, genuinely underexplored dimension of this broader dietary picture, one that recent advances in ancient microbiome research have only just begun to illuminate. Researchers analyzing preserved dental calculus and, in rarer cases, coprolite samples for traces of ancient microbial DNA have identified bacterial species within Neanderthal samples that closely resemble strains found in the modern human gut microbiome, associated with efficient digestion of both animal protein and complex plant carbohydrates, evidence suggesting Neanderthal digestive systems, much like their modern human counterparts, hosted a genuinely mixed, omnivore-adapted microbial community rather than the more specialized gut flora, typical of obligate carnivores, a finding that provides yet another independent line of biological evidence reinforcing the broader dietary flexibility. This reconstruction has documented through isotopes, dental calculus, coprolites, and competitive energetics alike. This microbiome evidence carries a further, genuinely striking implication, since a gut microbial community adapted to efficiently process both meat and plant carbohydrate would itself have represented a meaningful nutritional resource in its own right, allowing Neanderthal populations to extract usable energy and nutrients from a broader range of available foods than a more narrowly specialized digestive system could have managed. A biological flexibility operating at the microscopic, bacterial level that complements and helps explain the behavioral and dietary flexibility already documented at the level of individual archaeological sites scattered from Gibraltar to Siberia. The full weight of this evidence, gathered piece by piece across more than a decade of increasingly sophisticated isotopic, microscopic, and biomolecular research, points toward a conclusion that would have seemed genuinely surprising to researchers working even 20 years ago, when the simple, unified image of Neanderthals as pure, specialized, big game hunters still dominated the scientific literature almost without serious challenge. Surviving the Ice Age, it turns out, required considerably more dietary ingenuity, regional adaptability, and outright nutritional problem-solving than that older, simpler picture ever allowed for. A population meeting the same basic caloric and physiological demands, roughly 4,000 kilocalories a day, and a careful, deliberate balance between protein and fat intake, through an impressively wide range of locally tailored solutions, each one representing generations of accumulated, transmitted, place-specific knowledge about exactly which animals to hunt, which plants to dig, which bones to crush for their hidden fat, and, in at least one remarkable, still debated case from northern Spain, which bitter, unpalatable plants might actually help when the body itself began to fail, too.